right, so that's an awesome talk on meiotic drive, and I'll continue the trend, and then there's one more meiotic drive talk after me, so a nice little trio of meiotic drive talks. So even though this is an evolution conference, I have a slight disclaimer, which is that this is really just a simple genetics talk, because my main goal is to convince you that that is true. And the evolutionary implications of that, which I think are interesting, I'll sort of leave to your imaginations and questions if there's time. And that's the fun part anyway. Meiosis is obviously a major cellular event in the life cycle, and that's because it's producing these haploid gametes that then fuse and resume the diploid phase of the life cycle. So we all know that. But it's also a pivotal event because in a lot of lineages, it's intrinsically creating the conditions for competition and conflict over transmission. And that's because as a reductive division, haploid or homologous chromosomes are segregating into their own gametes, and those homologous chromosomes are often genetically distinct from each other. And just as not all gametes achieve fertilization, sort of the realm of sexual selection, not all gametes need even be transmitted. And so the ingredients for natural selection are present within organisms during meiosis and gametogenesis themselves. And so this creates selection for alleles that can bias their own transmission at the expense of the alternate allele, which is now sitting on a homologous chromosome in another gamete. And this biased transmission has been found in a wide range of organisms and broadly defined is called meiotic drive. And what's fascinating is that this selection is acting on the machinery of meiosis itself. And so really in these lineages, to understand meiosis, we need to understand the distribution and the frequency and the evolutionary dynamics and the mechanism of meiotic drive. So there's been a lot of work on this, and one thing that's really clear is that how meiotic drive occurs really depends on how meiosis itself occurs. And in seed plants and animals, meiosis is very different between males and females. So in males, meiosis is symmetrical, and so after the 2 meiotic divisions, there are 4 meiotic products that can all transmit genetic material. And so to achieve drive in males, a drive allele has to be quite destructive, and it has to prevent the development of sperm that are carrying, or pollen, carrying the alternate allele. In contrast, female meiosis is asymmetric, and only one of the meiotic products transmits genetic material in the first place, and the other 3 degrade as polar bodies. And so one way to achieve drive in females is just to end up in the egg. And what's really fascinating is that one of the loci that has been shown to be to cause female drive is the centromere itself. And so mechanistic work in mice has shown that stronger centromeres are able to preferentially orient themselves towards the egg. And basically what this does is it's relegating the weaker centromere, or the non-drive allele, towards the polar body. And so even though these mechanisms are quite different, what they share in common is that they're being selected for during meiosis and not because they're contributing to a phenotype. And they should spread rapidly because of that. And so the quite large differences in centromeres, for example, between species is thought to be because of this form of selection. So one subclass of male meiotic drive is when the driver is linked to an X chromosome. And so what happens here, sort of there's some unique consequences to sex linkage, is that the sperm that are getting disrupted and not being transmitted are those that carry the Y chromosome. And so this leads to sex ratio distortion in the offspring. And this is a phenotype that really has facilitated the discovery of these. And in fact, the first meiotic driver that was discovered almost 100 years ago by this Russian scientist in Drosophila was one of these X-linked drivers. And there's been, I think, 15 to 20, new ones keep getting discovered of these in Drosophila alone, and a spattering of other organisms as well. And so one of the unique consequences of sex linkage is as these spread through populations, they lead to a sex distortion at the population level, and eventually, in theory, extinction due to a lack of males. But in these species of Drosophila, what we often see is that the populations are polymorphic, and so there's a wild-type chromosome and a driving chromosome. And so there's been a lot of work characterizing the forces that are preventing the spread of a driver. So the driver is doing well in meiosis and it spreads, but then there are, for example, fitness consequences of carrying the driver that lead to polymorphism. So we recently discovered one of these drivers in this fly, Drosophila testacea. It's widespread across Europe and Asia, but we have predominantly studied a population in Switzerland. And in this population, there are two types of males, wild-type males that produce 50-50 sex ratios, and driving males that produce predominantly female offspring. And just as in a lot of other systems, the driving chromosome is actually at fairly low frequency. And so about 5 to 10% of males carry the driving X chromosome. And so, like I said, in a lot of systems, people have done a lot of work to characterize the forces preventing the spread, and we see it at low frequency. And so we also wanted to know, what is counteracting the spread of the driving chromosome? And so low-hanging fruit is to look at the fitness of the different genotypes. And so we performed this cross where you take a heterozygous female, cross it to a wild-type male, and you get these sons that are genetically identical apart from their X chromosome. And then you can do various fitness experiments, fertility, longevity, or what have you. So over the years, we've done a number of these experiments, and we noticed a bizarre pattern, which is that over time, we just noticed that, there was always an excess of driving males. And we kind of scratched our head for a while why that was the case, and it eventually dawned on us that maybe there was distortion occurring through females. So we wanted to formally test that, and so Steve Perlman, which is my master's supervisor, underwent this Herculean task of isolating a phenotypic marker. This is not Drosophila melanogaster, so there are no genetic resources, but through screening tens of thousands of flies, he isolated this phenotypic marker that's on the X chromosome, and it gives these flies a bright eye phenotype. So now we have two non-driving chromosomes. One is wild-type eye color, one with the bright eye color. And then we still have the driving chromosome, which has the wild-type eye color. And so you can produce these females that are heterozygous for a driving X chromosome and a non-driving X chromosome with the bright eye. And the bright eye is recessive, so these females have the wild-type eye color. So if you cross these to a bright eye male, you get all of the offspring that have the wild-type eye color have inherited the driving X chromosome from their mom, and all of the offspring with the bright eye phenotype have inherited the non-driving bright eye chromosome from their mom. So this is a very simple way to basically compete the chromosomes through the female, and just by quantifying the proportion of eye color in their offspring. And what's nice is we still have the fully wild-type X chromosome, and so we can do a similar cross, pairing the two wild-type, or the two non-driving chromosomes with each other. And these chromosomes only differ in that bright eye mutation, and so this should lead to just the equal Mendelian proportion of eye color in the offspring. And so the world is a logical place, and so that control cross gave us perfectly Mendelian transmission of the two X chromosomes. But the world is also a very bizarre place because we found significant distortion of the driving X chromosome, which is it's a sperm-killing male mitotic-drive chromosome, and here it is distorting transmission through females. So 64% on average of these females, or 64% of their offspring had the wild-type eye color, and therefore 64% had inherited the driving X chromosome from the mom. And we found this across 3 independently collected lines from the wild, and so this distortion is out there in the wild. So we found that there's distortion among adults, and we wanted to know when during the life cycle this distortion is arising. And obviously we were very interested in whether this was occurring during meiosis. So we want to get as close to meiosis as we can, and that means we wanted to see the proportion of these two chromosomes in eggs. So we performed the same cross and collected embryos, and embryos don't have eyes, so we need a different way of genotyping them. And so the two X chromosomes turn out to be quite divergent from one another, and so we could develop PCR markers that differentiate the two X chromosomes, and so allowing us to genotype embryos. And across, colors don't work, that's okay. The taller bars are the driving X chromosomes. Across 3 females, on average, we found 64% of the embryos had inherited the driving X chromosome. 138 out of 214 embryos had inherited the driving X chromosome, and therefore this distortion that we saw among adults was already present in eggs and had occurred prezygotically. And so it is an example of female meiotic drive. And so this chromosome is able to distort meiosis in both males and females, which are two fundamentally different processes of meiosis. And there are many examples of chromosomes or alleles that can drive in males and alone, and other examples of drive in females alone, but this is the first example of a chromosome that can distort through both. So this obviously has a number of implications for the evolutionary dynamics of this driving X chromosome. You know, there's questions about what came first, male drive or female drive, and of course questions about how these are distorting in the first place. And so to get at sort of the last two, we collaborated with Benjamin Lopin and his PhD student Emmanuel, and they produced these karyotypes for us. And so shown with the white arrows is the non-driving wild-type chromosome. It's an acrocentric X chromosome. And really nothing could have prepared me for what the driver looks like because it's absolutely massive. It's twice the size of the wild-type chromosome. And it sort of stains a little brighter, which suggests that it's heterochromatic. And it's on the centromeric side. And so we don't really know how it's causing drive, but one sort of tantalizing hypothesis is that it involves a fairly massively expanded centromere. And so we're doing some genomics and trying to figure out what that crazy heterochromatic arm actually looks like and how that might inform a mechanism. So with that, I'd like to thank everyone that was involved and you for listening.
Okay, I'll just start. So my name's Sasha. I'm a PhD student at University College London, and we've already had a really great introduction to meiotic drive, so you've made my job really easy, especially because I think this is a little bit of a sidestep maybe to the talks that we've had. But today I'm going to be talking to you about the energetic and metabolic costs of meiotic drive in this really cool species. So if you don't know what this is a stalk-eyed fly, or Teleopsis dalmani. So they're really well known for their characteristic sexual selection. They have these really crazy, exaggerated eye stalks. A lot is known in terms of the kind of secondary sexual ornaments, and the trade-offs of these is a big feature in our lab that we like to look at. But they're also really well known for having X-linked meiotic drive. So I don't need to introduce you to this now, but in case anyone new has joined the room, essentially it's just a selfish genetic element that biases normal meiosis in a way in which, especially when present on the sex chromosomes, might also cause a sex ratio bias. Now, where drive genes are found is not random. They tend to be in areas of low recombination, such as within inversions, but that means that they might accumulate deleterious mutations that could manifest as costs to the host. So in my system, like I said, we have this X-linked distorter, and its effect in males kills all the Y-bearing sperm, some of the Y-bearing sperm, to create these female biased broods. Now, this idea of fitness costs associated with drive has been a huge motivator for research in my lab. Particularly, I'm quite interested in how these different traits and phenotypes manifest. So a lot of the work which has been done out of this system has typically been on maintaining male fertility, despite the obvious deficit of half of the sperm being killed. I'll just highlight my own work here. So we found that the males with the drive genotype have evolved much, much larger testes in early development to compensate for this effect of sperm killing. So as a consequence, they're able to produce adequate levels of sperm comparative to wild type. So we've looked at the sperm count, so even per ejaculate, the exact same number of sperm is transferred to the females in comparison to wild type, despite half of them initially being killed. So this is a kind of novel putative adaptation, I suppose, to compensate for some of the effects of drive. And some of the other research that has been found tends to be looking at this being traded off with having both smaller eye span and smaller accessory glands as well, which are the components responsible for all the non-sperm components of the seminal fluid, and also the remating rate. So lots of work has been done so far. Oh, let me go back. Is this the right one? There we go. So lots of work has been done so far on the morphological and reproductive traits in this species between drive and standard. But myself and one of my supervisors, Flo Camus, we were thinking maybe what about the metabolic traits? What about the energetic traits? You know, we hadn't really seen much research linking this with meiotic drive before. So we thought, why not investigate this a little bit more? But before I get into the experiments, I just wanted to define the genotypes that are being used here. So we have obviously standard males and standard females. I'll refer to them as ST throughout. And then we also have drive individuals referred to as SR throughout, which stands for sex ratio, as you already know. And because there's only 2 male genotypes and sometimes 3 female genotypes, I like to pull the hets and homs together to make a direct comparison with males, so I'll say when I do that. So a natural first place to look at, if we wanted to discover the metabolic traits a little bit more, is to look at the mitochondria. So a really quick reminder of the electron transport chain. There are 4 main protein complexes which pump protons across the membrane to promote the formation of ADP into ATP by ATP synthase, or complex 5, during oxidative phosphorylation. And we analyzed the mitochondrial function using the Ouroboros, which we dissected the samples, the thoracic tissue, we weighed them, and then they were mechanically permeabilized. And we had no protocols for this. We spent a lot of time doing the protocol optimization for this as well. So then when you get it, something like this comes out. I won't go into that too much. But essentially, you just supply the different complexes with different substrates that activate them all sequentially. You can repress them as well to look in fine detail at those different processes during OXPHOS. But I guess the main important term that you need to know is just the respiratory control ratio, or RCR. Now, this is a standard measurement that is always looked at in this sort of research. So basically, this just shows how efficient oxidative phosphorylation is. So a high RCR implies mitochondria have a high capacity for substrate oxidation and high ADP to ATP turnover, as well as a low protein leak. And we found that the genotype significantly affects the RCR. Importantly, the ST individuals had a larger RCR than the SR individuals. So they were shown to have a better coupled or more efficient respiration in the thoracic tissue. There was no apparent difference between the sexes here. And I'll just mention here that we didn't actually look at heterozygotes in the mitochondrial experiments. Secondly, I'll also show you this complex, so complex 1. Again, in this sort of research, complex 1 tends to be the main thing that people look at. It's the biggest complex with the most subunits. It's vital for redox homeostasis in cells, so that's why I draw particular attention to it here. And we also find that genotype, again, significantly affects the percentage of complex 1 contribution to respiration. So ST individuals display a higher percentage of this complex's contribution to respiration, but again, we don't see any difference across the sexes. So just to summarize that experiment, we found that drive or SR individuals have this lowered RCR and lowered phosphorylation capacity. SR individuals have a reduced percentage contribution of C1 to respiration. So there's some evidence so far in this early stages that drive alters mitochondrial function, but it's unknown actually from these whether there's an additive effect of the drive allele, because we didn't test heterozygotes in this experiment. Now, the second experiment to go along with this looks at measuring whole organism metabolic rate. So this is called a MAVEN, and essentially you have a fly in each of those little chambers in the middle. They receive this airflow in succession. The live animal respires as normal. The air is pushed out to an external gas analyzer, and then it measures the amount of CO2 in each chamber. So what you're getting is the second-by-second activity of a live individual respiring over a several-hour cycle. So the way that we like to look at this is the more CO2 out is a higher respiration, but it's less efficient. So less CO2 out, there's less respiration, and it's more efficient. So they have a lower basal metabolic rate, which is better. And we know that respiration can be affected by lots of different factors. So weight, age, activity level. Is it affected by this genotype as well? And again, I just list all the genotypes here at the bottom that we tested in these experiments. And we found that respiration differed significantly between the SR and the ST genotypes, where SR had a greater basal metabolic rate than standards. Respiration differed between the sexes as well, but we didn't see any genotype-sex interaction. And then just to compare directly, so I mentioned, we can't directly compare drive with females because of the difference in the number of the genotypes. But when we pull them, we found that, again, standard has lower than both SR hets and HOMs, but there's actually no difference between females with either one or two chromosomes that we can see here. So just to summarize that experiment, SR individuals have this higher basal metabolic rate than ST individuals. One or two SR copy number in females makes no difference here. And drive individuals are expelling more CO2. So are they using oxygen less efficiently, which was one of the things we were thinking about? And then could we, in theory, link this back to potentially the mitochondria being impaired? So the final experiment that we wanted to do, just to kind of complete the story, was to look at consumption. So again, this is a really well-known assay called the CAFE assay, developed in Ben's lab. So essentially, we wanted to test them on 3 different diets. So these are different diets to what we would normally feed them on. So we had to do lots of trial work to see whether they would actually eat it in the first place. So we have this higher protein, kind of middle or lower protein, and then a sugar solution. And again, we're testing those 5 different genotype groups. So essentially, the way this works is that little thing sticking out at the top is a glass capillary. You uptake the liquid diet in the capillary, and then the meniscus will decline by loss of because of the loss of solution, both by evaporation and consumption. So obviously, we also have controls just to measure how much is naturally lost through evaporation. We replaced and measured the food differences at 20 hours, 40 hours, and 60 hours. So only total consumption is shown here, so that difference across the whole time. And here we have this. So we found that drive, again, SR individuals, they consume more than ST individuals. We don't see a difference in sex here, but I suppose this makes sense because they're all controlled for body size anyway. We know that diet significantly affected consumption. Again, that's not particularly crazy, but we don't normally feed them a diet rich in protein, so we wanted to see if this had any effect anyway. And we, again, see a preference for higher protein compared with lower protein compared with sugar. When I looked just within the females, we saw that true genotype, by true genotype I mean whether it was heterozygous, homozygous, it didn't significantly affect the consumption, apart from we see this difference in het females consuming more than the others. But we think this is probably just a problem with power analysis. But either way, we still see that the diet remains a significant attributor to consumption within females. But again, you do generally see this difference with drive individuals consuming more. So just to summarize that final experiment, we found that drive individuals consume more than standard individuals. We don't see any sex differences here on consumption. Diets with higher protein are preferred. Potentially, are they needing extra nutrients for inefficient metabolism? So just to discuss this as one story, if you have inefficient oxidative phosphorylation due to some kind of alteration or impairment with the mitochondria, if you think back to the Krebs cycle, you're making less ATP per molecule of substrate supplied. So therefore, you will need more substrate required to make the same amount of ATP, and then the byproduct of that is you're expelling even more CO2. So if we link this to the experiments that we've looked at, we have this low rate of Coupling, so there's low rate of coupling within the ETC, so there's low RCR in drive individuals. There's definitely something going on there with the drive genotypes with regards to the mitochondria. They're eating a lot more. Potentially, they've got this inability to sufficiently utilize nutrients in the body. And again, we also see this compared with the fact that they have this kind of less efficient respiration. So if you remember at the start of my talk, I mentioned that we know there's multiple inversions spanning this X chromosome that we look at in this system. Now, a lot of our other work has been based on the fact that we suspect there to be an accumulation of deleterious mutations that manifest as these costly traits in the host. Now, we now have a genome that's been put together, so we can actually begin to determine the kind of genetic underpinnings of this. But I think this is a good place to start with regards to looking for maybe some candidate genes potentially that are interrupting proteins that are involved in complex 1 that might actually be impairing the mitochondria here. But for now, I think this is a first kind of good step that tends to look at the metabolic and kind of energetic traits that might be associated with host fitness for the hosts that are harboring drive. So I'll just leave you with that, and I'll say thank you to my different supervisors, POM, Flo, lots of some students and undergrads who've been really helpful in the gathering of this work. And then also just a little plug to say that I'm looking for a postdoc at the start of next year. So yeah, if you just got any cool ideas that you want to discuss with me, I'm interested in lots of different things, all through sexual selection, mate choice, gametes. I love lots of different stuff. So if you want to come and have a chat with me in the coffee break, I'd love to speak with you. And thank you all for listening.
All right, thanks for coming, and thanks to the organizers. I'm going to talk about a new project in my lab on B chromosomes, and this work was done by two people, primarily Paul Ginsberg, there on the left, who's a postdoc, and Evelyn Queller, who's an undergraduate and looking for a tech job in California, if anybody's hiring. So a little bit of background on B chromosomes. B chromosomes are non-essential and common. They're present in something like 15% of eukaryotic species. This is a case in Drosophila melanogaster, and this is sort of a lab-derived B chromosome. It's caused by a mutation on on the fourth chromosome, and it sort of spits off all these small dot chromosomes. Can you see my pointer? No, you can't see anything. Okay. And we see these in other species as well. There's maize, so those pink spots are the chromosomes that are bees, and also silver foxes, and so on. And B chromosome spread and persistence is generally thought to be in this sort of conflict scenario because they're not necessary. So they might drive. And so this is two flavors of drive. There's lots of ways a B chromosome could drive. But one way they could drive is they could preferentially get themselves into the germline instead of the soma. And another way is to do female drive, like what Graeme talked about, and preferentially get into the egg instead of the polar bodies. And the assumption is that this usually comes with some fitness cost. So there's something maintaining some intermediate frequency of these B chromosomes. And a fitness cost is a good guess. It's also a good guess that that fitness cost increases with copy number of Bs. So this is a case from a cricket where the increasing B copy number leads to decreased fitness. And so this is one of those stories that I saw when I was a grad student in this book. So Patterson and Stone, and if you study Drosophila, especially if you study non-melanogaster Drosophila, this book is like the Bible. And it has incredible amounts of data from 1952, but still totally relevant today. And I saw these two karyotypes for Drosophila putrida. I'll give you more information on Drosophila putrida in a minute. And what I was interested in is there was a sort of standard Drosophila karyotype on the left, and then this different one on the right. And there are a couple of things here. So first of all, there's this extra chromosome, and this looks like a dot chromosome. There's the the dot, sorry, that looks like the B chromosome. There's the dot chromosome, which is a standard feature of a Drosophila genome. And then the other thing that's kind of interesting here is that Y chromosome on the left is a lot longer than the one on the right. So that was intriguing to me. This is Drosophila putrida. It's a widespread mushroom feeder, so those black dots there are all the locales that it was collected in this particular study. And it's a member of the Testacea group. So the story that Graeme told you, this is a species that's sort of the outgroup of that Testacea species group. And just for context, that's 50 to 70 million years diverged from Melanogaster. So what we wanted to do is just basically get our first characterization of this. So we wanted to re-isolate the bee. It hadn't been studied since the 40s and 50s. We wanted to look at frequency in natural populations. We wanted to look at maintenance. And then we wanted to study the origin. Where did it come from? Okay, so here's our initial Patterson and Stone plots. And what I'll show you is a squash from females, one female that had no bees, one female that had bees, and that's this. So let's walk through what we're looking at here. So we have X chromosomes. I don't know if that's way too small over there. I'm going to zoom in on this in a second. We have we have the dot chromosomes. And then we have bees on the right, not on the left. Looks similar in males. We have an X and a Y, of course. There's our dot, and there's our bees. Just for reference, the female up here has 3, male has 2 B chromosomes. So there's the zoom in where you can see it a little bit better. 2 X's, the dots, and the B's. What's interesting here is the dot is actually big compared to what we usually see in a chromosome. Okay, this is a lot of work put into one slide and ignoring a lot of detail. This is the frequency across several populations in the eastern part of the United States. And so B positive is orange, and you can see B positive is fairly frequent, especially in those populations where we've got decent sample sizes. This plot is a little bit complicated. So this is the number of bees for each isofemale line from each of those populations. So the dot here is the mean for that isofemale line, and the error bar is interline variation. So there is interline variation. And then the violin is sort of the distribution for the entire population. So that's the Florida population, and here's the rest of them. And so you can see there's a lot of variation within each of these locations, but fairly consistent among the populations, at least with the power we have with these sample sizes. So the next thing we wanted to look at, so the B chromosome exists, it's common. Next thing we wanted to do is look at how it's maintained. And so the first thing we wanted to look at is fitness. And this is a relatively crude fitness assay, but basically we made B replacement lines. So we have lines that are isogenic except for the presence or absence of the B chromosome. And we crossed in all possible combinations of B plus female, B minus male, and so on. We let them lay eggs for 7 days, and we just counted adults. Fairly crude measure of fitness. There's much more precise ways to do this. And the data looks something like this. So on the y-axis is total offspring produced, and then you have each of the different cross types on the x-axis. So both negative, mom negative, dad positive, mom positive, dad negative, and positive, positive. And it looks something like this. So not a huge effect, and a lot of noise. And if we do an ANOVA, there's no effect of mom, there's no effect of dad, and there's no effect of the interaction between mom and dad. But if we ignore statistics, not that I'm suggesting you do that, the B minus moms do produce a lot less offspring. And so if you just calculate that cost based on ignoring statistics, it's a huge cost. So we clearly need to do something different in terms of measuring the cost, but maybe there's something in these females. So let's move on to transmission. So again, B replacement lines, and we're doing all the pairwise combinations. Of course, we don't expect any transmission in the B minus to B minus cross. But what we do here is we extract DNA from both parents and from all the offspring, and we're going to do droplet digital PCR. And I'm not going to go into a lot of the details here, but droplet digital PCR is going to basically give us a ratio of a specific B locus to an autosomal locus. And so we can measure that as the relative amount of B chromosome that we have. I should say a couple things here. One is that in our B positive line, there is variation. So we have to check each individual that we do with this approach. The other thing is we took the offspring and we broke them into 4 samples wherever we could. 2 female and 2 male samples that are pools of a bunch of males and females. So there's going to be variation among those just because these are relatively small numbers and it's just sampling. So this is what the data looks like. Transmission is on the y-axis here, and the expectation for transmission is a little bit odd, so let me explain that. The expectation is if you have 1 B chromosome, you transmit it to half of your offspring. Think about a Y chromosome. It's the same sort of idea. If you have 2 B chromosomes, you should transmit it to everybody, and so on. So we're normalizing to the expectation. Whatever the parents had, we're normalizing back to that expectation. So this is what it looks like for moms that are B minus and dads that are B plus. And so it's transmitting through dads, and there's a significant increase above that expectation, 0.02. For B plus moms, it's a lot more spread, but it's also a significant increase above that expectation. I should say all the different colors and shapes here are just the different sexes and their different error bars, but you can kind of just zoom out and look. We don't see an effect of which sex we're looking at. And then B plus by B plus, interestingly, we don't see a significant transmission effect. We need some more data here. That might be because we start to get to these fitness costs if we're getting lots of B chromosomes, but it's too early to say anything definitive about that. Okay, so it looks like there's some drive, maybe some fitness costs, but you're gonna have to come back and hear the next talk on that. The next thing we wanted to do is look at what is the B chromosome, what's the origin of it? And to do that, we sequenced B plus and B minus female, of both sexes with PacBio, and we just turned the standard crank where we're looking at coverage with both PacBio and Illumina data across the different scaffolds to try to find things that are B-linked, and it looks something like this. So scaffold 1 on the top there, the coverage for the B plus line is about the same as the B minus line, so that's probably not a B chromosome. But scaffold 2, the B plus line, has much higher coverage than B minus, so that might be a B chromosome. It looks like this. So all those things in reddish, orangish are enriched for coverage in the B line. And we can look at that in another way, which is this, which is just coverage across one of these scaffolds. And you can see much higher coverage in the B, which is red, than in the B minus, which is blue. And then we can do PCR to just say, yes, this is actually B-linked. It only amplifies in those B lines. So what's on it? This is where fly people might be more excited than the rest of you. The main thing that's on it is this canonical Y-linked gene in Drosophila called KL2. And KL2 is required for fertility. Now, we think this gene is still on most of the Y chromosomes in the wild as well. And quickly, it's spread throughout this entire scaffold. So this is mapping on the x-axis the amino acid from the KL2 in Drosophila virilis to the position in the scaffold where it maps in, on the putrid B chromosome. So it's got these huge 100+ kb introns, which is typical of a Y-linked gene, so this all kind of makes sense. And then last, we looked at the repeats on this B chromosome, and briefly, this is all the Y chromosome here. On the left, you can see all the different repeat classes and whether they're Y-specific or they're everywhere, or if they were X-specific or anything like that. And either they're repeats that are everywhere or they're just Y-specific, and that's it. Okay, so what I've shown you is that these B chromosomes are there, they're common, and they vary in copy numbers 0 to 4. There may be some fitness cost through female, but certainly noisy. Looks like it's driving through both sexes, so that's another thing that we're seeing in common today. And the origin is likely the Y chromosome with maybe some traffic. The last thing I want to leave you with is there's this evidence. So when I showed you those karyotypes at the beginning, I said one of the Ys looked like it was shorter. And I thought, well, maybe if it broke off to form the B chromosome and that Y was still segregating the populations, now you have this possibility that if that gene is essential for fertility, you could lose it and not have not be fertile. And so we screened all these wild males and found a couple of males that were sterile, and we couldn't amplify KL2. So that suggests that maybe that's true, but that needs some more work as well. Okay, I'm finished. I want to quickly put a plug. KU is hiring lots of postdocs, my lab included. If you're interested, come talk to me. I want to thank a lot of people, but mostly Paul and Evelyn and the organizers of the symposium. And I think I might not have time for questions, but thank you.
Good afternoon, everyone. It's almost afternoon, and thanks for the organizers for having me here. So I'm currently at UT San Antonio. That's my new home. I just started there. Today I'll mostly talk about stuff that I did as a postdoc at Sloan Kettering Institute. And we have heard a lot of meiotic drive talks today. That's very exciting, and I've learned a lot today. But in this talk, I'll try to focus mostly on the suppression side of meiotic drive and how RNA molecules can be involved in suppressing meiotic drive genes in using Drosophila simulans male germline as a model. So an organism's genome is like a parliament of genes. We've heard this metaphor already once today, and it's inspired from Lay's Adaptation and Diversity book. So in a parliament, people often have to come together to make important decisions for the well-being of the country. Then parliament also has people who have voices of dissent. You may have already realized this. If you haven't, the next time a spending bill comes, regardless of which country you are from, you will realize how dysfunctional the parliamentary setup is. The same happens in the genome. So there are genes, many genes that perform critical cellular and developmental functions that are essential for the organisms. Now, these are genes that are deeply conserved across organisms. The genome is also home to genes that can be selfish, and this can be transposable elements, which make the bulk of many eukaryotic organisms, and also meiotic drive genes that violate Mendelian segregation. Now, in general, these selfish genes are detrimental to host reproductive fitness, and sort of this talk of difference in interest between the host and these selfish genes triggers genetic conflicts and consequently evolve mechanisms of defense against meiotic drive. So we've heard a lot about meiotic drive introduction, so I'll not dwell much on this. So meiotic drive is genetic cheating where alleles violate this fair segregation of alleles. But in addition to that, what I want to point out here is that the true extent of how much meiotic drive happens in nature is not very well known, partly because in many instances, meiotic drive may not have any overt phenotypes. So if I were to replace this big A and little a with, let's say, sex chromosomes, then there is a phenotypic readout of this meiotic drive, which is sex chromosome drive, where there's imbalance in transmission of X and Y seen as sex ratio distortion in the progeny. I'll just focus on two examples here, and I'll talk about one example in Drosophila in detail. And the reason why I've mentioned these two examples is that where a driver is mapped, or at least we know the identity of the drivers. I mean, we've heard about talks this morning about meiotic drive in different systems where we know the drive is happening, but the identities of the drivers themselves are not known. So in the system that I'll focus today, it's called the distorter on the X system. This was mapped from work by Yun Tao, where the drive results in sex ratio bias. And I also have a mouse example here, partly because when I talk to my molecular biology colleagues, I often get this question that, is this a weird thing in insights? No, like, look, this is your major vertebrate model organism, and you have meiotic drive here. And in the mouse system, the genes that cause meiotic drive are called the SLX and SLY genes. So these are recent duplicates of synaptonormal complex genes that are involved in recombination that are massively amplified on the X and Y chromosome. And a stoichiometric balance of these genes are required to maintain a 50-50 sex ratio. And if the balance is lost, then it'll skew the sex ratio in the progeny. So the system that I'll focus today is Drosophila simulans, which is a sister species to the model fly Drosophila melanogaster. And in Drosophila simulans, genetic work championed by Yun Tao identified a driver on the X chromosome called the distorter on X, or DOX. Now this was a gene that was cloned and sequenced in simulants at that time. And when Yun did a simple blast search, Yun was not able to find a homologous sequence in Drosophila melanogaster. It was assumed that this DOX is a novel gene that emerged in simulants. However, in the genetic study, Yun was also able to find that there was another paralogous gene on the X chromosome called mother of DOX, and it was presumed that the DOX gene emerged from this MDOX. And interestingly, the genetic study also characterized a suppressor. In this case, I'm showing in autosome as A. It's called not much yang, or NIMI. And interestingly, this locus is a retropost copy of the driver itself. On the autosome, it got duplicated and exists as an inverted repeat region in the genome. So loss of NIMI results in a sex ratio bias phenotype. So given the structure, inverted repeat structure, we wondered whether the NIMI is an endogenous RNAi generating locus and whether the suppression is achieved by RNAi silencing. So this is the structure of NIMI. So NIMI is a nice inverted repeat. And these repetitive structures are nice substrates for this enzyme called endonuclease called dicer2, which can process these inverted repeats into small RNAs of size 21 nucleotides. I mean, if anyone in the audience, if you've done RNAi experiments, knockdown experiments, so this is the pathway that you'll feed your double-stranded RNA molecules into. But in this case, this is a naturally occurring double-stranded repeat in the genome. So what happens then is that these small RNAs are loaded onto organoid complexes. So these are silencing complexes that can then target the DOX mRNA using complementarity and destroy them. So if this is the model by which silencing is occurring, then it would make a prediction that if we were to make a mutant of the NIMI locus, then we would see derepression of the DOX region. So what I'm showing here is that we made CRISPR mutants of the NIMI inverted repeat in Drosophila simulans and replaced it with a DS-RED marker. And let me pull up my marker. It's in French. Okay, so what you're seeing here is the left is the WXT1, which is a wild-type phenotype. You can see barely any expression of the DOX gene. But in the NIMI mutant, you see not just DOX, but also the mother of DOX gene is massively de-repressed in a NIMI mutant. Now, this is all good that it recapitulated the genetic results, but then using PacBio genome in Drosophila simulans, we also discovered 2 additional paralogs of DOX family genes, which we call them the paradox genes, and also another inverted repeat structure on the autosome, which is very close to NIMI, roughly a megabase away from NIMI, called too much yin. And in fact, like another of Yun's genetic studies mapped TIMI as a suppressor of an unknown driver in simulants, and we found that the TIMI is also another inverted repeat. Now, why have multiple redundant suppressors here? So there are 2 possibilities here. Maybe like 2 suppressors evolved to co-silence different targets, DOX targets on the X chromosome, or there is some kind of target specificity here which would indicate recurrent bouts of drive and suppression happen in Drosophila simulans. So to distinguish these possibilities, we made a CRISPR knockout of the TIMI hairpin RNA and replaced it with a ubiquitin GFP marker. And what you can see here is in a TME mutant, the rightmost panel in the qPCR bar is that we specifically see the PDOX genes derepressed in a TME mutant, whereas DOX and MDOX genes in the NEME mutant, indicating there is targeting specificity for these APN RNAs to different DOX family members. So what this suggests is that the drive systems, particularly the PDOX drive and the DOX drive systems, have recurrently emerged via recurrent bouts of drive and suppression and simulants, which is kind of also recapitulated in our phenotypic assays, where we see distinct spermogenic phenotypes in both deletion of NEME and TME mutants, where in NEME mutants we see sex ratio distortion, whereas deleting the TME APN RNA leads to male sterility. Now, and again, like if you think about this, none of these genes and suppressors exist in the closest sister species, Drosophila melanogaster. And this made us wonder, like, where did this DOX gene come from, and what is it related to? So when we looked at the DOX gene more carefully, and we profiled transcriptomes from multiple tissues and Drosophila simulants, and what we found was the DOX is specifically expressed in the testis. And we also found that DOX is of chimeric origin. It's a fusion of at least 3 different genes, and probably that's the reason why the initial blast searches failed to identify an ortholog in Drosophila melanogaster. But what we found was the open reading frame of DOX contained a domain of unknown function called DUF1074. So using both sequence homology and also structural guided homology with AlphaFold 2, we found that the domain of unknown function 1074 is actually an HMG box domain. So this is a domain that is found in many transcription factors and also in sperm nuclear basic proteins that are involved in histone to protamine transition. And we found that the DOX gene, the open reading frame, is more closely related to a gene called protamine. So what do protamines do? So after meiosis in spermatogenesis, the haploid spermatids, which are round in shape, undergo a large scale chromatin remodification, where the histones that wrap the DNA are replaced by these sperm nuclear basic proteins called protamines, which gives the sperm this nice compact needle-like structure. And this compaction is achieved greater than 200-fold with protamines replacing histones. And this histone to protamine transition is also an evolutionarily conserved mechanism. And although the fly protamines and mouse protamines are not orthologs, suggesting the protamine-based packaging has evolved multiple times. Now, this provided us some clues to how these the protamine mimics can derail spermatogenesis, and although mechanistically we still don't have a clear picture of how DOX can derail spermatogenesis. So far what I've showed you today is the emergence of multiple DOX family drivers on the X chromosome and consequent emergence of suppressors by means of APN RNAs to silence these genes. But what I haven't discussed is whether the DOX factors are truly causal for sex ratio distortion and sterility. Now, a very straightforward, and in my mind, a clean experiment to show causal relationship would be to just do CRISPR of individual DOX family genes and see if we could rescue that phenotype. And we've tried that experiment so many times, but it failed. Unfortunately, we were not able to get any CRISPR mutant. But fortunately for us, when we surveyed polymorphisms of DOX genes across different Drosophila simulant strains, we found naturally occurring deletions at the DOX family loci in Drosophila simulants. So I'm just showing an example, 2 strains here, MD15 and NS40, both from Africa, that have natural deletions at the DOX region. So now if DOX is causal for this phenotype, then in these strains, if we were to put them in a sensitized background, that is in a NEME, a DEL background, deletion background, then we should see rescue of sex ratio distortion. And we did this experiment in collaboration with Colin Michael-John at the University of Nebraska. And what we found that the naturally occurring deletions at the DOX family loci can rescue sex ratio distortion. So the first in the panel is the wild type flies with a functional copy of DOX, where you can see 95 to 99% sex ratio, which is completely rescued in strains lacking the DOX gene. Same thing as what we observed with the PDOX locus. there are naturally occurring PDOX deletions in Drosophila simulans, the NS40 strain, and we are also able to rescue sterility in that strain. A lack of PDOX2 can rescue sterility, suggesting that the DOX family genes are indeed causal for the phenotype that we are observing. Now, this polymorphism within simulants motivated us to go look at the DOX family genes in more detail in the simulants clade, and what we found was the DOX family genes emerged in the ancestor to the simulants clade. We also detect DOX genes in Drosophila mauritiana and Seychellia, the sister species to Drosophila simulans. And today I showed two suppressors that evolved to silence DOX family genes, NIMI and TIMI. And on the X chromosome in Drosophila mauritiana and Seychellia, these DOX genes have massively amplified. Together we detected 22 copies, and which also led to the emergence of novel TIMI-like suppressors in Mauritiana, which is called TIMI2, and another TIMI tandem cluster in Drosophila Seychellia. So showing us that this sort of tit-for-tat coevolution with an expanding cohort of drivers on the X chromosome, which are then suppressed by epNRNA suppressors. Now we are interested in characterizing the molecular mechanisms of how drive is mediated, and also in my own independent group at UTSA, we are interested in examining the breadth of RNAi usage in suppressing meiotic drive in both Drosophilids and beyond. So with that, I'd like to thank, the bulk of the work was done as a postdoc at Sloan Kettering Institute, so I'd like to thank Eric Lai and my collaborations at Sloan Kettering, a former graduate student, Gillian Lin with the CRISPR mutants, and also Benjamin Lepon's lab in France, and Colin Michael John at University of Nebraska. So we are a new group at UTSA, and if you're interested in genetic conflicts, and like me, if you're interested in doing both experiments and bioinformatics, please get in touch. We can discuss this. And I'm happy to take questions.
So, hi everyone. My name is Fanny Mazzamurro. I'm a PhD student in the microbial evolutionary genomics unit at the Pasteur Institute in Paris, and I'm working on the evolution of natural transformation in bacteria. So, natural transformation was first observed in 1928 by Frederick Griffiths during one of his experiments. So, in his experiments, you had a virulent strain. He killed a virulent strain, which was thus rendered non-virulent, a non-virulent strain. And what he observed is that when he mixed the non-virulent strain with the heat-killed virulent strain, he was able to recover the virulent strain from the blood of the dead mouse. And so he hypothesized at the time that there was a transforming principle passing from the heat-killed virulent strain to the non-virulent strain. And a little bit less than 20 years later, Avery and his collaborators identified this transforming principle as being DNA. So today, natural transformation is one of the only mechanisms of horizontal gene transfer in bacteria that is under the control of the recipient bacteria. So it consists in the uptake of exogenous DNA, its integration in the bacterial chromosome by homologous recombination. Several evolutionary hypotheses have been proposed to explain the existence of natural transformation, one of them being that the uptaken DNA might be used as a source of nutrient for the recipient bacteria. But by homologous recombination, transformation might also help repair damaged DNA, break genetic linkage, and also help in the acquisition or the loss of genes. But a more recent hypothesis, which is called the chromosome curing model, suggests that transformation may cure the bacterial chromosome from selfish genetic elements, such as mobile genetic elements. But in reaction to such a cleaning, mobile genetic elements might defend themselves by impairing transformation. So large variation in the natural transformation rate had been observed between bacterial species, but also within bacterial species. So we wanted to characterize the large variation that exists in these rates, try to identify the genetic determinants, and test the different theories I've just presented to you. So to do so, we worked with two bacterial species, Legionella pneumophila and Acinetobacter baumannii. So the first step was to check if there were indeed variations to work on. So this was done by one of our collaborators who performed a transformation luminescence assay So in this type of assay, the luminescence is used as a proxy for the transformation rate. And thus we obtain those two distributions of transformation rate in Acetobacter baumannii and Legionella. Since we are interested in distinguishing between transformable and non-transformable strains, we had to set a threshold, and it was based on the maximum of the distribution of the transformation rate of a non-transformable strain. And with this, we ended up with 64% of the Acetobacter baumannii collection, which was transformable, and 52% in Legionella. So it's nice to have this kind of distribution, but it was more interesting for us to know if closely related strains could actually share the same phenotype. So we built the recombination-free phylogenetic tree in both collections, and we plotted on them the transformation rates, but we also plotted as a continuous measure, but also the binary phenotype. And when you plot the binary phenotype, you don't see any obvious pattern or cluster of transferable or non-transferable strain. So now that we have been introduced to the two collections, we can test the different evolutionary hypotheses. So we started by the chromosome curing model, and we actually found several arguments in favor of the impairment of natural transformation by mobigenetic elements. So when we were looking, when we tried to identify the genetic determinants of the variation in natural transformation rates, we performed the genome-wide association study on the binary phenotype, and we obtained this kind of plot. So here it's for Acetobacter baumannii. Each circle represents a gene family. On the x-axis, you have the effect size. If the effect size is negative, then the gene is associated with the inhibition of transformation. But if it's, the effect size is positive, then it is associated with transformability. On the y-axis, you have the significancy. So the higher, the more significant the association is. In this graph, all the genes that are over the red threshold are considered significantly associated. And as we can see, it's a lot of genes. So instead of investigating each and every gene, and since we were interested in mobigenetic elements impairing transformation, we decided to color the gene by the mobigenetic elements they belong to. So we had characterized and identified different types of mobigenetic elements. So we color them by prophage. As you can see, it is associated with the inhibition of transformation in Acetobacter baumannii. We also color them whether or not they were carrying by an integrative conjugative elements, by a plasmid, or by an insertion sequences. And we did the same in Legionella pneumophila. And this time, the most striking inhibition of transformation was found in the plasmid category. So in both species, you can see that mobigenetic elements seem to have an inhibiting role of the transformability. Another argument in favor of the impairment of transformation by mobigenetic elements was found when we decided to model the evolution of the transformation rate. So we fitted different evolutionary models. We started obviously by the most classic one, which is the Brownian motion model. And actually the fit wasn't very good. And the best fit we obtained was when we fitted the data with the jump process model. So in a jump process, the trait, so here the transformation trait, changed suddenly over a short period of time. And this could be consistent with mobigenetic elements being acquired or lost suddenly. So now that we have an idea that the phenotype evolved by jumps, we wanted to see if we could actually observe jumps that cause phenotype change in the tree. So we ancestrally reconstructed the binary transformation phenotype, and we only focused on the recent events and the terminal branches. In these terminal branches, we were able to observe phenotype changes, and we actually noticed that there was an excess of changes towards non-transformability compared to transformability. So this can have this was true in both species, and this could be interpreted in two ways. Either in the long term, it means that the transferable strains are doomed, but actually we are currently observing half of the collection being transferable, so it's very unlikely. The second hypothesis, which is more likely, is that the non-transferable strains are actually counter-selected on the long term, and that's why we see this pattern. So here I'm just summarizing all the arguments that we found in favor of the mobile genetic elements impairing transformation. So this is only the evolutionary consequence of the chromosome-curing model, but what about the essence of the model, which is that transformation removes mobile genetic elements? So to test if it was actually true, we counted the number of, to simplify, the number of conjugative systems and insertion sequences in the different phenotypes, and we saw that there were less conjugative systems and less insertion sequences in transformable strains, which goes in favor of the transformation curing the bacterial chromosome. But we are also interested in the defense systems that could interact with biologic elements. So we divided them in two. First, the innate defense systems, which are not CRISPR-Cas systems, such as restriction modification systems. So these systems were actually more present in non-transformable strains, and this makes sense since this type of system are able to block the entry of the uptake, not the uptake, the uptaken DNA in the bacteria. On the contrary, CRISPR-Cas systems were more present in transformable strains, and this can be explained by the fact that CRISPR-Cas can impair the entry of phages and plasmids, and so potentially mobile genetic elements that inhibits transformation. Another hypothesis we could and we wanted to test was whether or not transformation was indeed breaking genetic linkage. To be able to do this, transformation has to have good homologous recombination systems. And so we first checked whether or not there were higher recombination rates in transformable strain compared to non-transformable ones, and actually it was the case. And then to estimate genetic linkage, we measured the linkage disequilibrium, and we found that there was a lower linkage disequilibrium in transformable strains compared to non-transformable ones. So this is in favor of transformation breaking down genetic linkage. The last hypothesis we'd like to test, and we're currently investigating, is the role of transformation in gene flow. So whether transformation favors the acquisition or the loss of genes. And to do so, we are going to rely on three main features, which is the number of genes, which here does not seem to depend on the transformation phenotype, the number of acquisition events, and the balance between gene gains and gene losses. So to conclude, I hope I've shown you that there are striking parallels between the two bacterial species in the evolution of the natural transformation rates. In both species, the transformation rates were widely variable, and this could be due to the evolutionary conflicts that exist between mobile genetic elements and transformation. But we should not forget that natural transformation has not a unique evolutionary role. It plays a role in the decrease in genetic linkage, but also in gene flow. And with this, I'd just like to thank my supervisor, my collaborators, my lab, and you for your attention.
Okay, thanks very much for the opportunities to speak here, the organizers. I'm going to talk, this will be a bit different. This is talking about synthetic systems rather than naturally occurring systems. And the motivating force here is the idea of using synthetic genetic constructs for pest control. Ooh. All right. Okay, it's not moving here, so I'm going to move here so I can see. Yeah, okay, fine. Okay, so yeah, so this is about genetic pest control, and the idea here is that if you want to control a pest species, you release modified members of that pest species in order to control it. The simple, most widespread example of this is releasing irradiation sterilized males to control a pest population that's been used for decades now in some agricultural systems, limiting screwworm from North America, and it's used actively for medfly control, et cetera. So it has the potential to be an environmentally friendly, species-specific, quite effective mechanism approach for certain species, and the number of species where you can use this is kind of limited at the moment, but their recent advances in synthetic biology and molecular biology are opening up the toolbox of what might be possible using for genetic biocontrol. If you think about a developmental pipeline for developing a strain like this, typically you want to see if it works well on the computer, then you might see if it works in the lab, and then eventually see if it works in the field. This talk is purely about the first step, what seems to work well in the computer. So we take two different extremes here. So we've got, on the one hand, the release of sterile males. So that requires repeated releases of males to suppress the population. They'll mate with the females don't have progeny, and so the population is suppressed. And that gives, that will suppress the population in the area where you release those males, but not elsewhere. And that can work in some cases, but it's impractical if your target species per target population is very large, or if the species is difficult to rear, because you have to inundate the population. You have to release a lot. At the other extreme, there are ideas about using gene drive. We've heard a lot about gene drive this morning, naturally occurring systems, but synthetic gene drive systems to control a population. And there, instead of inundating a population, you're inoculating it. So you release relatively few individuals, and then over successive generations, the construct will spread through the population, and if it's knocking out a female fertility gene, then it will suppress and eliminate the population. But that has the property that because it can spread from relatively small numbers, it will also spread away from the area in which you release it. And so that's okay if the species you're targeting is harmful everywhere it exists. But if you want to target your control into a specific population, then that sort of approach is going to be inappropriate. So what's in between those two extremes, where we have something that's more efficient than sterile males, but not as spreadable as gene drive? So if we look at the allele frequencies of those two ideas, if you're releasing a sterile male after one generation, those sterile male, that's whatever is responsible for the sterility has disappeared from the population, and that's why you have to keep doing repeated releases. On the other hand, if you've got gene drive spreading, then that you release it, and then over successive generations, that increases in frequency. So what's in between those two possibilities is something that just maintains and persists at whatever frequency you put it in at. So that's the sort of thing we are wondering about how we might engineer that. There have been two previous designs or proposals about how to control populations using more or less things that will persist, more or less selectively neutral constructs. One is to put a construct on a Y chromosome that will make mutations on, so this is an XY male species, for example. So you put the construct on the Y chromosome such that it makes mutations such that the daughters of those males then are lethal or sterile. So, for example, making dominant mutations on the X chromosome. So that will kill the females, the daughters, but the Y chromosome itself is not affected by that because the Y chromosome is not in those daughters. And so its frequency can be maintained. Another way of doing it is to make dominant female sterile or lethal mutations, which, so the construct is dominant female sterile and shows strong drive in males. And so you can get, if you have strong drive in males and dominant lethality in females, then you can get a balance there. And that could be done, for example, by targeting a female-specific haploinsufficient gene. So both of these can show under idealized circumstances neutral dynamics, and then otherwise with imperfect efficiencies will just persist and slowly disappear. So here I'm going to describe a third design which has similar sorts of properties, but it doesn't require dealing with sex chromosomes and it doesn't require sex-specific effects. And it is using the notion of drive selection balance. So if you want to suppress a population, you might, the obvious thing to do is to start maybe knocking out a gene that's involved in female, forget female, a gene that's involved in survival or reproduction. So an essential gene. So selection then, if you're knocking out that gene, selection would normally remove that allele, that knockout from the population. Most knockouts of essential genes are recessive, and that means that selection against those recessives is weak when the gene is rare in the population. As the gene becomes more and more common in the population, then selection gets stronger. And so that's the orange line there. That's the relative fitness of the construct. It's close to 1 when it's very rare, and then close and very small as it gets common. So we want, if we're going to do drive selection balance, we want a form of drive that will sort of mirror that selection, the effect of selection. And so there are toxin antidote forms of drive which do exactly that. So if you've got a gene which in a heterozygote is transmitted to 50% of the progeny, but the 50% of the progeny that don't inherit that gene die or are sterile, then that can give the gene an advantage. It's a very weak advantage when the construct is rare, but it's a strong advantage when the construct is common. And so mathematically, under the ideal circumstances, it exactly mirrors that selection against a recessive gene, a recessive knockout gene of an essential gene. So these two are perfectly balanced under the ideals. And despite that having a neutral fitness, so they're going to show neutral dynamics, it can impose a strong reproductive load upon the population, which increases with its frequency. When it's rare, a small reproductive load, but as it gets common, the load gets higher and higher. And so that's going to load here being the proportion of the reproductive output of the population that is destroyed, either because the individual is homozygous for the knockout or because it's making this toxin without the antidote. Okay, so to make this a little more concrete, how could you imagine building such a thing? Well, start with the drive part of it. We have a CRISPR-Cas9 with a guide RNA on somewhere in the genome that is targeting a haploinsufficient gene elsewhere in the genome, so making mutations in it. Then you have a recoded version of that haploinsufficient gene next to your CRISPR-Cas9 construct. So that protects that construct from the fact that the other copies are mutated, but only that copy. And you stick that whole thing in the middle of a haplosufficient gene, an essential gene, knocking out that haplosufficient gene to make the recessive lethality. So, and we call this a protected dominant negative editor, pDNA. There's in some ways, simpler way, perhaps doing it with just a single locus rather than two loci where you don't have to recode anything. If you can work with a gene that is haplosufficient, so knockouts are recessive lethal, but you are able to edit it, to make an edit, that is a dominant lethal. And so if that's downstream of where you're introducing your construct, then that can work. And so just to give you an example, there's if you look at flybase, there's 45 genes in Drosophila that, where there's both recessive lethal and dominant lethal mutations known. So these sorts of things are not impossible. Okay, so how does this kind of construct compare to others? So here we're imagining, this is just some simulations showing what happens if you do 10% releases successfully, generation after generation. And you can see that the purple line there shows the RPDNE, and it is pretty much as good as a Y-linked editor, and substantially better than the other sorts of proposals that have been made, such as an X shredder, or Riddle, or the sterile insect technique. And if you just look at the release rates required to get, for example, 95% suppression of the population in 36 generations, you need to do releases of an SIT about 130% of your initial target population, and so it goes down. It's only 4% every generation releases for the Y-linked editor and the pDNA. So substantially more efficient. All right, we can also, those are under ideal circumstances. Things won't be ideal, of course. We can also look at the sensitivity of the efficacy due to changes in the different parameters, the editing rates, the homozygous fitness effects, et cetera. Won't have time to go through those, but they're more or less as one would expect. We also would want to know, is there any way in which this driver, this construct could turn into a driver? And if we're promising it to stay localized, could something happen that it wouldn't be? And so it turns out that it's quite unlikely as long as you're inserting this thing into an essential gene such that the knockout is a homozygous lethal, then that's not going to be possible. And it's also stable against the different kind of deletion derivatives that might arise once you release it. And just very briefly, this thing could be made even more efficient just by adding elsewhere in the genome a guide RNA that would allow your construct to home in the presence of that guide RNA. So that's kind of giving it a booster. So instead of doing repeated releases, you could imagine controlling and eliminating a population just with a single release. And that's what the bottom plot shows, where just a single 100% release, the pDNA continues to go up after that because it's being boosted by this booster, which is encoding for the homing reaction. So summary then, new tools are needed for genetic biocontrol, particularly strategies that combine high efficiencies but still localization. We're proposing a design here of using a recessive lethal that produces dominant lethals, edits killing the progeny that don't inherit the construct. Under idealized circumstances, this is going to show the selection is precisely balanced by the drive, and so this behaves as if selectively neutral despite imposing a large load, reproductive load. I kind of think of this as analogous to a perpetual motion machine. In reality, perfect balance like perpetual motion is not likely to happen, not going to be possible, but even with some imperfections, the design can be highly efficient suppression. And it's these inevitable imperfections actually which is going to mean the thing is localized and not spread without limit over a landscape. And further improvements can be made by having this booster, which allows for single control with just a single release rather than repeated releases. And just acknowledgements, this was done, this work was done with a postdoc in the lab, Katie Willis, and it's done under the auspices of the Target Malaria Program, which is a not-for-profit research consortium developing new tools for malaria control with core funding from the Gates Foundation, an open philanthropy project. And there's a preprint on this on bioRxiv. Thanks very much.